Good evening, and welcome to our public lecture series sponsored by the Center for STEM Excellence. Sacramento State's Center for STEM Excellence works with area schools, colleges, universities, and employers to attract and educate young people for careers in science, technology, engineering, and mathematics. My name is Jill Traynor, and I'm Dean of the College of Natural Sciences and Mathematics and co-director for the Center for STEM Excellence. Let me take this opportunity to invite you to our next lecture in the public lecture series, which will be on April 19th at 6 o'clock, same time, in this room, the Redwood Room, same place. The speaker, Dr. Donald Cohn, director of the UCLA David Geffen School of Medicine Human Gene Medicine Program, will be speaking about regenerative medicine, stem cell medicine. Tonight, it is my pleasure to introduce our speaker, Dr. Samantha Hens. Since the year 2000, Dr. Hens has been a faculty member in Sacramento State's Department of Anthropology. She received her BA in anthropology from the State University of New York at Buffalo. She moved on to complete her MA and PhD in anthropology at the University of Tennessee, Knoxville. Dr. Hens held a postdoctoral position at Johns Hopkins University School of Medicine, where she began using three-dimensional geomorphic morphometric techniques. She's used these techniques to study the development of sexual dimorphism in orangutans and gorillas. And more recently, she's turned her interest in three-dimensional analysis of growth and development to past human populations, particularly individuals from ancient Rome. Dr. Hens balances her active research program with her love of teaching, earning the College of Social Sciences and Interdisciplinary Studies Outstanding Teacher Award in 2006. Please join me in welcoming Dr. Samantha Hens.
Good evening. I'd like to just start by welcoming everybody and thanking you so very much for coming. I was very honored to have been asked to give tonight's presentation and have the ability to speak to you this evening, and the turnout is fantastic. So I invited a lot of family and friends just in case, nobody else showed up. So I'm very happy to see all of you. Let's see, will this work? We seem to not want to advance. This may take a second. There we go. Hopefully this will be our only glitch. I'd like to begin with just a brief overview, as you can see there, of the structure of the talk. We'll begin with a brief description of biological anthropology so that the members of the audience may gain an understanding of what it is that we actually do and my background. From there, we'll move on to how biological anthropologists have traditionally studied health in past populations, and we'll hopefully see some interesting examples of some pathological lesions. That's the two approaches to health there. After that, I'd like to introduce you to the study of asymmetry and what it can teach us about health during development or developmental health. And then with this groundwork completed, we'll move on to a case study using asymmetry as a measure of population health in Imperial Romans there. And I'd like to conclude with just a short discussion, 5 minutes or so, on the importance of the Center for STEM Excellence and what I feel it has to do with something called the unity of knowledge. It's working, okay. Anthropology is a holistic field, encompassing the study of humans from both biological and cultural standpoints. And it accomplishes this by looking at populations from both the past and the current day. The subfield of anthropology, traditionally called physical or biological anthropology, studies the place of humans across both space and time. These anthropologists strive to unite the biological information that they gather about humans, things like skeletal structure or genetic information, with our understanding of the cultural processes that shape our evolution. So some biological anthropologists examine our evolutionary past, as you can see here, through the discovery and interpretation of fossil ancestors or fossil remains. Others down here, right? That's not an anthropologist, of course, that's a gorilla. But others explore our place within the primate order so that we can understand how living primates can inform us about our own behavior and adaptation. Some, like myself, prefer the study of human skeletal remains to understand health, disease, growth, and demography in past populations, and then some specialize in the health of modern populations. All of these fields of inquiry just brush the surface of the kind of work undertaken by biological anthropologists today, but hopefully they provide a basic scope of the field. My concern here today is how a branch of biological anthropology known as human skeletal biology approaches and answers questions about the health, growth, and development of ancient populations. Traditionally, to study health in past populations, one would adopt one of two approaches. This first approach, we're documenting standard measurements on the skeletal remains of past peoples and comparing those measurements to standards for normal health and growth. These are two growth curves for children. What you're seeing here is this is the humerus for the upper bone of the arm, and you'll notice this line, the darker one down the center, is the average or the median, and then the lines on either side represent the range and variation. So here on the y-axis, the vertical, we see the length of the humerus, and then down here, age in years from birth to 12. Similar type of thing is drawn here, but this one is with a line describing the length of the femur, or the thigh bone, and we see the length of the femur here, 40, 50, 60 millimeters, but in this case, this is for fetal remains, so it's taking us through 5 through 10 lunar months, if you wanted to determine the age of a very young individual. Both of these show a relationship between the age of the child and the length of the long bones, but both of them can also be used to compare long bone length to what we call a quote-unquote normal standard of development. So for example, children with poor health and poor nutrition are known to have shorter than average height compared to well-nourished or healthy children. This difference is reflected in the length of their limb bones. So let's say, for example, you were looking here and you measured the length of a humerus. If the child was 6, it's a little odd angle for me, but it looks like it's about 180 millimeters or 18 centimeters there. If you came up with a child that was only 140 millimeters for the length of the humerus and it was a 6-year-old child, you would say that this child is a little bit underdeveloped, smaller than average. Many studies have demonstrated this difference between groups of people from disparate backgrounds. In the second approach, we know that many traumatic and pathological or disease processes leave their mark on bone. So one could record the frequency of these lesions on bone and relate it to the health of the people. And honestly, this is the most popular and standard approach. Here, what you're looking at are two fractured femora. These are thigh bones. This one's just laying on its side, and this one is vertical. And they also both show healing, which followed the injury. What you're seeing here, obviously this is unset, but all of this extra bone growth that's kind of curling over the edges indicates the bone continued growing and trying to heal itself after the fracture. Here, you'll notice that even though it's not set, these are pre-modern populations, that the bone is much smoother. So it's something called a fracture callus. This indicates a bone that's been long healed. This one, the person unlikely did not survive the injury. These are both Civil War era soldiers. So most pathological lesions that we find can be classified into 3 groups: abnormal bone loss, excess bone formation, and deformity. So I'd like to present a few of the more commonly seen pathologies for your enjoyment. Okay. The first one is Porotic Hyperostosis. This is a disease linked to iron deficiency anemia. Mainly, it's seen in the bones of the vault or the brain case in particular, and they respond by making more red blood cells, which causes the skull bones to expand and become porous. That's what these arrows are showing here. This is the top of a skull, and those are the bones coming together at the sutures. And you see all of these pores. This is what it should look like, nice and smooth. This is a very common location for these lesions. Here is the back of somebody's skull. The big spaces there are just areas where small bones have fallen out, but we're looking at all these small pores across the back of the head. Here you see this excess growth in the eye orbits, and it would be a related problem. Archaeologically, we commonly see this condition in populations with nutritional deficiencies in iron, often due to over-reliance on crops that inhibit iron absorption, or sometimes in areas where genetic anemias are common, and those are usually linked to malaria. Okay. The next one, degenerative joint disease or osteoarthritis, one that many of us are familiar with or will be familiar with as we age. This involves the breakdown of the joint surfaces and, as I mentioned, has been linked to normal aging, but also to hard labor or the overuse of certain joints. So for example, if you were a baseball player and for years, were pitching with that same arm and you have a lot of overuse in the shoulder and in the elbow, you may see these osteoarthritic changes in those regions. Here you're seeing the breakdown of the joint surface along with bony protrusions. So this is a hip, this is the bone of the thigh, and then the pelvis, and this ball here would fit into that socket. And you notice that there's kind of a rim or a ring that should not be there. And this socket is extra large and kind of overgrown. This is the knee joint, the bone of the thigh and the shin. These edges of these bones, these surfaces should be very smooth, and instead you see, I kind of describe them as cruddy looking, but all this extra bone growth and formation. These are the 5 vertebrae in your lower back, the lumbar vertebrae. Each of these bones here should be separated and nice and distinct, and that is indeed not what you're seeing. You're noticing that there's a lot of overgrowth. In some cases, there's fusion of the actual vertebrae to each other, which would completely inhibit movement. This is a very severe case of arthritis in the lower back. Archaeologically, degenerative joint disease teaches us a lot about sex-specific roles within a community. Often males and females will have varying frequencies and locations of degenerative changes, and that can indicate different lifestyles or tasks based on your sex role in the society. Osteomyelitis, my students all know this one's my favorite. This is a very serious condition, and it results from an infection occurring inside the marrow cavity of the bone. The bone eventually dies if it's not treated, and then new, rather haphazard, chaotic bone in terms of its organization is laid down on top of the infected area, and there's drainage holes for any necrotic material. This is a femur or thigh bone. Down here you have the bone of the upper arm, the humerus, and these are the 2 bones in your lower leg, your tibia and fibula, the shin. So what you're seeing here, obviously this is not a nice smooth surface, even the way it looked much better even in the fractured bone. All of this excess bone formation. Same thing here. You can see the original shaft underneath, and then there's rather a sleeve that's been built up around it. And you're seeing the same thing here as these fuse together. These holes that you're seeing there are for drainage of the necrotic material. These are very commonly seen in war-torn areas, but they're not only archaeological, they're very much a serious condition today. These could result from a staph infection that got into the bone if you were in the hospital. These images in particular come from, again, injured Civil War soldiers, and all of these would have resulted in the amputation of the limb. These are all very severe. Treponemal infection is caused by a microorganism that invades the bone, and it is very commonly seen in the skull. What you're looking at here is the top of the head, and then over here looking at a side view, and this bone here should be very smooth. It should be very flat. Same thing with here on the side. You'll notice that there's this kind of eroded look to it. It's often been described as worm-eaten or snail track, which is kind of what describes these lesions. They're very characteristic. In tropical populations, which is what you're seeing here, it's often manifested in the lower legs, between the knee and the ankle. And you can see in this individual swelling in the feet, but also that kind of anterior posterior bowing of the legs, which is an unusual, they should be very straight there. In tropical populations, the reason it manifests often in the lower legs is the microorganism is usually picked up out of the soil through like cuts or scrapes in the feet or ankles. However, when you get to temperate climates, which is usually where you're seeing it in the skull, the area, the microorganism needs a warm, moist area, and it migrates to the genital region, and it's known as venereal syphilis. So there's much debate archaeologically in terms of the origin and spread of this treponemal disease between the New and Old World. Basically, did Columbus bring it here, or did we give it to Columbus? That kind of thing. Tuberculosis is another microorganism that in its later stages may invade the vertebrae of the back. The bones become riddled with holes and may eventually collapse, creating a hunchback appearance. What you're seeing here is an up-close view of the vertebrae in the center of your back, the thoracic ones that you can see the ribs there, which have been cut off. But you'll notice that they kind of look like Swiss cheese, which they should not. They should have a nice smooth surface. And this one in particular, the holes were so bad that it's actually fractured right in half. And what you're looking at with both of these, which are specimens from the Armed Forces Institute of Pathology in Washington, D.C., these vertebrae above and below on both of these individuals look pretty nice. Those look very healthy. But then right in the middle is where the microorganism invaded, and it actually caused collapse, and you can see the hunchback that would result from this. This pathology is more common in urban areas with crowded, unsanitary conditions, but it has been discovered as far back as the ancient Egyptian mummies. Okay. The next one is Osteomalacia, which is more commonly known as rickets. This is due to a deficiency in vitamin D, and as you can see, it creates deformed long bones. These are all the bones of the lower limb. This is the thigh, and then the 2 bones of the lower leg. You can see that this obvious bowing, and this is in a kind of outside-inside, or what we call medial-lateral orientation, not normal, would give you the bowed leg appearance. This one, not so much bowed, but the head, the ball here is kind of a little bit too low and kind of tight and shortened, and overall the bone is rather short and stumpy. It should be much more long and slender. So again, that's a vitamin D deficiency. This is one that many of our audience will be familiar with at some point in their life. This is osteoporosis. This is an age-related thinning of the cortex, or the shaft of the bone. Excuse me, sorry about that. There we go. What we have here on the left, this is a femur or thigh bone, and it's been sawed in half so that we can see inside. And you'll notice the normal marrow cavity, and then you can see the thickness of the bone. This is a nice, healthy bone, showing the thickness that should exist. This is the same bone, different individual, but you'll notice how basically huge the marrow cavity is and how thin that shaft is around there, the cortical bone, as we say. So this is that age-related thinning. We see it very commonly in menopausal women. And it results, of course, in elderly women often falling and fracturing hips, wrists, as it may be. Okay, I had a little preview there almost. These are talk about dental health a little bit. What we're seeing here is a few different things, but the first thing to mention is dental caries, or what you would commonly call cavities. These are very common in populations after the development of agriculture. Generally very rare in pre-agricultural populations, or what we would call hunter-gatherers. It's the dietary intake of the refined carbohydrates and starches that's negatively impacting oral health. So you've got the roof of somebody's mouth here, and you can see the large carious lesions going on there, and also wherever you see these kind of dark dots. I don't have the steadiest hand, which is why I'm not in surgery, right? But those are also beginning early carious lesions. What you'll also notice here is this really large hole. There's a big space here where the bone's been eaten away. You see one here on this individual as well as up here. All of those are abscesses. Those are signs of pretty severe infection, and these could and probably did result in the death of these individuals. Another thing to notice here is you can see the roots of the teeth, which are slightly yellow compared to the enamel-covered crowns, which are white. You notice that the roots are visible above or below the jaw there. Normally, the bone would come right up to the junction of the enamel and the roots there, and it's drawn back. That's periodontal disease, and it's a good reminder to brush and floss before you go to bed or in the morning. Finally, the last one to mention under this heading is enamel hypoplasias. These have been intensively studied, and they're due to infection or malnutrition. And what happens is the cells that make the enamel in the fetus or the young child when the teeth are forming are actually killed off, and they stop laying down enamel. And this creates these lines that you'll see here, or bands or pits or grooves. They can be different forms. You'll see one here across this upper tooth, along with across the lower teeth there in these teeth. Archaeologically, the study of enamel hypoplasias helps us to understand childhood health as the enamel is formed, as I said, during these fetal and childhood stages. So even though we're seeing it on fully adult skulls here, it's telling us about what happened in their childhood, not really what's happening currently. Enamel hypoplasias are a measure of stress during development. So anthropologists have studied the frequency of these lesions for several decades as an indicator of population health. All of the diseases that I've shown here have been identified in past populations across the globe. In most cases, they've been linked to the health and well-being of these people, including their growth, nutritional deficiencies, dietary differences, but also things like work habits, migration patterns, or differences due to sex and status. So for example, like I said earlier, males and females might differ in the amount of osteoarthritis in their joints, as well as the location of the arthritic areas. And that can tell us quite a bit about the activities in which each sex was participating. And so while all of these are very fascinating, and as I said, take up the bulk of literature in health and disease in ancient populations, the work I'd like to present today actually takes a quite different approach, and you might even think of it as a third approach to studying health. Okay. Symmetry is the idea that the left and right sides of the body are symmetrical or mirror images of one another, and it has many applications throughout biology and clinical medicine. Researchers have identified symmetry as an important indicator of good health and survival prospects, even for humans. Now, obviously, not a human here, very simple artistic rendition showing you what a symmetric structure would look like versus an asymmetric structure. People with more symmetric faces are generally found attractive, and they may have a better chance of success at reproduction and genetic survival. Also, severe asymmetry may indicate genetic problems, or that an individual suffered from infections that affected its development, and that could suggest to potential mates that they might want to look elsewhere for a healthier partner. Some asymmetries have been linked to problems during growth and development due to genetic factors, but also poor nutrition and disease processes. Symmetry may be measured in numerous structures, including the bones, the teeth, or the soft tissue. There are three main types of asymmetry that I'd like to discuss: directional asymmetry, antisymmetry, and fluctuating asymmetry, which is why we're here today. So directional asymmetry, this occurs when there's greater development of a structure on one side of the body. It's very, very common in humans when you think of right and left-handedness. A right-handed individual will have longer limb dimensions on the right side, along with stronger musculature on the right side compared to the left. The mammalian heart would be another example. The left side is generally larger than the right side. So generally, directional asymmetry is related to genetic factors, and it's not related to any kind of stress during development. Here, what you're seeing are two femora, or thigh bones. These are from the same person, and it's easy to see that the left, this is the left bone for the person, it's on your right, but this left bone is obviously significantly longer than the one on the right. This is an example of directional asymmetry. This is also common where most humans are right-handed. We're also left leg dominant, so often the left leg dimensions will be larger than the right side. Antisymmetry is the case where asymmetry is present in all individuals in the population, but it's completely random whether it's going to be the right side or the left side is larger. It's not very commonly studied because it is somewhat rare, but the best example comes here. This is the signaling claw of the male fiddler crab. One claw is always going to be larger than the other, but it could be the right or the left, and it's completely random. It has a strong genetic basis, and it's not related to the health of the crab. There we go. Fluctuating asymmetry. These are quite a bit more complicated. These are very small, subtle, random deviations of normally symmetrical structures. They're associated with instability during development. This instability could be due to a numerous number of factors: temperature extremes, noise, pollution, nutritional deficiencies, population density, infection or disease, amongst many others. All of these are collectively often called developmental noise, basically the noise, these factors that are affecting you while you're trying to develop. So these numerous environmental factors can affect an individual's growth and development, and they can cause these random deviations from absolute perfect symmetry. Here you see a photo of a young girl. They've been digitally altered so that one shows a perfectly symmetrical feature, and the other, features I should say, and the other shows very minor variation. And I had to study these for quite a while until I finally figured it out. It helps, of course, if you put a piece of paper or a line down the center of her face. This young lady on the left is perfectly symmetrical, mirror image on the left and right sides. This one on the right is slightly asymmetrical, and you'll probably, most easiest to notice it is in the region of the eye where her left eye sits just a little bit higher than her right eye. Let's see here. So when we identify fluctuating asymmetry in an organism's physical traits, it's considered a reflection of environmental stress that happened during development, and that created this developmental instability and these fluctuations. So they are an indicator of the health of a population. Genetic studies have shown that fluctuating asymmetry has little or no genetic basis. However, with that said, individuals do vary in their ability to withstand such environmental stresses. So we do see population-level differences in resistance to developmental stress. So in other words, the ability of an organism to buffer itself against such environmental stressors during development has a strong genetic basis. So while the asymmetry itself does not have a genetic basis, your ability to buffer yourself against the stress while you're developing does have a genetic factor. Organisms strive for stability during development and the ability to develop symmetrically and produce what we call a consistent phenotype or physical form or shape. Despite all of this noise coming at you is something called canalization. So canalization acts as a stabilizing force, and it maintains symmetry, and it keeps the physical form consistent despite any disturbances. So people that have good, strong canalization, that's a genetic thing, right? And they have that going for them, and it helps them combat the environmental noise that's affecting them. So, as I mentioned, these destabilizing forces or these disturbances can be poor prenatal and maternal conditions. They can be related to socioeconomic level, disease, parasite load, or even toxin exposure. So here, I very bravely put up a photo of myself with bangs, which are now back in fashion, even though this is an old photo. This is normally an asymmetrical face, and it's a little bit easier to see, I think, because I drew this blue line through the center, and you can see that there are slight asymmetries between the left and the right side. Part of it's being picked up, of course, my angle of the face due to the camera. Put this in less technical terms, just to make sure you're all with me. Fluctuating asymmetry is a measure of an individual's genetic quality. The basic idea is that the genes within an individual should code for both sides of the body equally. So your left arm should develop like your right arm, your left eye like your right eye, your left ear like your right ear, and so on. But random events in the outside environment, like disease or poor nutrition, can interfere with this development. The better able an individual is to resist these outside influences, the better quality they and their genes are. They would be better canalized. So levels of fluctuating asymmetry in a population can inform us about the health of individuals, their genetic quality, and any disturbances that may have affected them during their growth and development. Let's see here. There we go. It also appears that levels of fluctuating asymmetry in a population can predict the survival prospects of the group. Research recently reported in the journal Conservation Biology suggests that thrush birds in Kenya, which you can see pictured here, an example of one of the species, they found that asymmetry increased the more degraded the forest fragment became. So asymmetry was 2.5 times higher in moderately disturbed forest regions compared to undisturbed regions, but it was 8 times higher when you had highly disturbed regions of the forest compared to undisturbed regions. And in these highly disturbed regions, the birds' survival rates were affected. They were dying sooner. So this suggests asymmetry does appear before significant damage is done to the population. Asymmetry, thus, could be used as an early danger sign and have real impact for conservation programs everywhere. The key, of course, is detecting it and recognizing that the population is under stress before any irreversible damage is done. So like these thrush birds from Kenya, there's been numerous animal models in clinical settings that have tested the relationship between fluctuating asymmetry and stress. Rats, for example, have been exposed to temperature variation, heat exposure, loud noise, protein deprivation. Some researchers have looked at the relationship between fluctuating asymmetry and maternal diabetes in rhesus macaques. And in all cases, the fluctuating asymmetry in the animals increased with exposure to these environmental stressors. The presence of parasites or a poor diet just compounds this because it affects an organism's ability to buffer against the stress. The research on human populations supports the idea that stressed populations have a higher level of fluctuating asymmetry. There's even been links demonstrated between fluctuating asymmetry in maternal smoking and fluctuating asymmetry in maternal obesity. The vast majority of research on humans has emphasized the dentition. There really hasn't been much attention at all paid to the skull, or what I would call the craniofacial skeleton, which is the face and the brain case. So at this time, we really don't understand if fluctuating asymmetry even affects the skull bones, and if it does, where these effects might appear. So in my research, I'm interested in determining whether or not we can detect fluctuating asymmetry in the craniofacial skeleton, and if so, what regions of the skull are more or less susceptible to these disturbances. And I found that the people of ancient Roman society really offered an interesting case study, along with the fact that it's very fun to work in Rome. So, the Roman imperial age is represented by archaeological sites across Italy, and indeed across a wider region than that. These skeletal populations allow us to examine health and stress among inhabitants of the Roman Empire. Many of the archaeological sites from the Imperial Roman era represent different socioeconomic regimes and provide an excellent opportunity to test hypotheses about the health and welfare of past populations and the effect of these environmental stressors on the body. The slide that you're seeing currently shows you some of the main archaeological sites available in Rome that represent the Imperial Roman era. I think almost everybody is going to be able to recognize the Colosseum. These two buildings here and in the lower right, this is the Temple of the Vestal Virgins. This is the Temple of Pius and Antonia. These are both structures that are found in the Roman Forum, and you can tour them today and get up close. This down here is just across the street from the Forum, and it's Trajan's Market. So again, structures from this region. In my research, I examined 3 Imperial Roman populations. The first one was the necropolis, which is a cemetery called Isola Sacra, and it was used by the inhabitants of the town of Portus, which is a coastal port city located along the coast about 30 kilometers west of Rome. You see it here on the map. You see Rome here, Florence, Milan, Venice, Naples. Isola Sacra and Portus are right there at the coast, just downriver from Rome. If you've ever traveled to the region or hope to, literally walking distance is also the site of Ostia Antica, and many of the pictures that I've used here are actually pictures from Ostia Antica, which is just a companion town walking distance from Portus. Same lifestyle, same type of issues, but sites that you can actually tour. Portus is not available today. Portus was an important port for trade and was closely linked to the social and economic life of the capital city of Rome. The Emperor Claudius began construction of the harbor in 41 AD, which was finished by the Emperor Trajan in 117 AD. Archaeological excavations here began in the 1920s and continued until the late 1980s, and they uncovered large monumental tombs along with numerous additional burials between the tombs. This photo here from the nearby site of Ostia Antica actually shows these small kind of openings, and these would have been places where you could place urns of cremated remains, but gives you an example of the type of sites that you see and the architecture of the region. Some more sites. The area was inhabited from the 1st to 3rd centuries AD and represents a population of urban, middle-class Romans. These are primarily craftsmen and shopkeepers. These people had continual access to resources due to their proximity to Rome and the location at a major trading port. Goods were routinely coming into the port and then being shipped upriver to Rome. The traditional diet of the Roman region emphasized cereals, along with wine, olives, lard, fruit, and dry legumes. Meat was actually a very minor component, and it was a luxury thought available more likely in the urban areas and especially for the wealthy. It would have been found, especially as pork would have been the most common source, and that would have been mostly ham and sausages, but there was also sheep, goats, and poultry eaten. Researchers have studied the chemical isotopes from the human remains at Isola Sacra and found that the inhabitants consumed a typical diet high in grains and other terrestrial foods, like I was saying, the olives and such, but also that they had significant access to marine resources. Due to the fact that they lived at the port, they were actually eating a lot of fish and shellfish, which is seriously increasing their protein component. What I have here is just sites of typical for that time period. This is a communal well, and you can see a very traditional Roman road existing off in the background. Romans were especially well known for their engineering feats. This is an apartment house, right? And you can see another road there going in front of it. And this is a mosaic, beautifully preserved. These were very, very common in front of stores or shops, where a lot of people, of course, could not read and write. Literacy was an issue at this time. And people, in order to sell their wares and their, would do these mosaics, advertising basically what they had. For example, we see some palm trees and a jar here, probably selling an oil, olive oil or palm oil, along with fish. This could have to do with the fact that they're selling fish or that they're involved in trade. The health and nutritional status of the individuals has also been examined using traditional paleopathological approaches, like I mentioned at the start of the talk. These are based on identification of these lesions that we saw. The presence of enamel hypoplasias suggests that the individuals from Portus experienced stress during their development. As long as they had moderate dental wear, they also had dental caries. This was an agricultural population. And they did have porotic hyperostosis, which were those pores in the crania. The porotic hyperostosis is very likely due to genetic anemias, which are common in the Mediterranean region due to the high malaria load. The second site was the site of Velia, and this was another necropolis that dates to the 1st to 2nd centuries AD, and it makes it contemporaneous with Isola Sacra. It's on the southwest coast of Italy, near the modern town of Salerno. So on this map, we're seeing it right there, south of Velia. This is another port trading center, and the inhabitants were traders, but they were especially employed in the construction and repair of ships. So I found 2 ship mosaics from the time to show you. They practiced small-scale cultivation or farming at this site. They fished the seas, and they operated a fish preservation industry. One of the most common ways that people would access fish was as kind of like a fish oil or a fish paste. It was something called garum that they would use in their cooking. An analysis of the chemical isotopes found in the bones from these people indicated a greater reliance on agricultural goods and cereals, with very limited consumption of meat and fish compared to the remains from Isola Sacra. So despite the fact that they've got this fish reservation industry, and despite the fact that they're a port town, they're actually not eating the fish. They're probably sending it elsewhere for trade. The final site that I studied was Castel Malnome. Very little is known about this site. It's been very recently excavated. We do know it dates between the 1st and 3rd centuries AD, making all 3 sites contemporaneous. And to be honest, they were literally cleaning the dirt off of the remains while I was in the lab. I often had to wait, kind of twiddle my thumbs for a while for a skull to be cleaned. The site, as you can see here, is very close to Isola Sacra and Portus, but it's actually inland, so it's not a coastal site. And it's near the modern-day town of Fiumicino, outside of modern-day Rome. The inhabitants here were involved in heavy labor associated with salt production, so salt miners. No research has been carried out on this population at this time, so I can't say much about their background. So Isola Sacra, Velia, and Castel Malnome are all members of the Imperial Roman era, but they all vary in their exposure to nutritional and other stresses. This is due in part to very different underlying economic conditions. So although the individuals buried at Velia and Isola Sacra are both from ports along the coast, their diets were significantly different. The inhabitants of Isola Sacra had a higher amount of meat and fish, and they experienced a more affluent lifestyle because they were traders and shopkeepers and craftsmen, while the people from Velia worked more in agriculture and manufacturing. The individuals from Castel Malnome, we know, represent an inland population of heavy laborers. They were likely, we estimate, represent a lower socioeconomic status, but we really don't know anything about their diet or anything at this point. So, before I move on, I'd like to give some recognition to my colleague on this project. Her name is Dr. Valerie DeLeon from Johns Hopkins University. While both of us are very familiar with asymmetry, Valerie was actually very familiar with the program used to analyze the data, and I could not have done this without her help. So at the start of the study, we predicted that Isola Sacra would be the healthiest population. They're along the major trading port. We know that they've got access to a lot of protein. They've got a middle class or affluent lifestyle. So we thought that they would show the least amount of fluctuating asymmetry, abbreviated that FA. We also anticipated that Castel Malnome would have the highest frequency of fluctuating asymmetry due to their lower socioeconomic status, and that way Velia would fall about in the middle with a moderate amount. So in our research, we examined fluctuating asymmetry in the craniofacial skeletons, the skulls, of all 3 populations. First, we compared Isola Sacra to Velia, the 2 coastal sites, and then Isola Sacra to Castel Malnome, since they were so close to each other in space, just one inland and one coastal. We did this using a very innovative and novel approach. We took 3-dimensional data from the skulls. Essentially, what we did was identify 34 landmarks or points on the skull. And you can see them all labeled there. The abbreviations are not so important, but you see that some of them are along the midline, and then you see the eye orbit, the sides of the face, along the top of the head, a bunch down here along the base of the skull, some in the midline, some off to the side. And then we used something called a 3-dimensional digitizer, which you can see pictured here, to actually capture the measurements. It's sort of a robotic arm, and it's very easy to use, actually. Digitizing is quite simple. It's the analysis which is the challenge. But this thing swivels and moves, and there's a stylus here, this little point which you can see resting, that you just put in your hand, and then you place this stylus along each point on the skull that you are interested in measuring. And when the stylus is there, you literally click a button, and it takes X, Y, Z coordinate data for each landmark. You're taking those, again, along the midline and along the side of the skull. So you're basically creating a three-dimensional representation of the skull. For those of you that are a little more mathematically inclined out there, we used a program called Euclidean Distance Matrix Analysis to calculate the distances and compare these. Basically, what we're doing is establishing the midline, which you can see here along the bottom and from the side and a little bit, or from the front there and a little bit from the side, and then we took these side landmarks. And what the program will do is, let's say this spot right here between the eyes stands for nasion. It'll measure this point in space and all the other points in space, and then it will calculate the distance between nasion and all other 33 landmarks, some of course in the midline, on each side. In this picture, I've only drawn them on the left just so that it wasn't a mass of too many labels. So let's see this point here. It'll take from BRG to every other point in space, from LMB to every other point in space. And it'll do this for both sides. So you get BRG to GWT on both the left and the right. You get BRG to EAU or to AST on both the left and the right. Same thing going on along the base of the skull. As you might imagine, what this is going to do is create a gargantuan matrix of numbers, and that's where the misery comes in for the analysts. This is going to be a 34 by 34 matrix of numbers, for those of you that have had matrix algebra. I'm not going to go into the very messy and complicated statistics here. One thing I will mention before I go on, though, the results were tested for statistical significance. So everything I present here in the results was found statistically significant. So again, we're comparing left and right sides for all possible dimensions. This is a very original approach compared to the approaches we saw earlier. These are not simple measurements just to see how long or short a bone is. They're not identifying lesions, which are kind of the end result, in most cases, what's happening to you at or near the time of death. We're doing something quite a bit different here. One, fluctuating asymmetry is showing us stress during development. So even though you're measuring it on an adult, it's showing you what happened while you were developing, while you were growing as a fetus and as a child. So you're not just seeing the lesions at the very tail end. Let's see here. Second, fluctuating asymmetry has been rarely studied in the adult human, in any human skull. So the fact that we're actually looking at it in the skull is also a novel approach. Third, this is the first three-dimensional analysis that's ever been carried out on imperial Roman area populations for any reason whatsoever, symmetry or otherwise. One of the nice things about this approach is that it incorporates a large number of characters. As you can see, these huge matrices of numbers into one single analysis. And what that means is it makes it likely that if there is asymmetry, we're going to be able to find it. We're going to be able to detect it. Another thing that's very significant is that it allows us to identify specific regions of the cranium that are most or least likely to show this developmental instability. So not only can we see how much asymmetry was in the population, but we can see where it's happening on the cranium. So we can identify potential patterns of asymmetry. So what did we find? Okay. The first analysis, as I said, we compared Isola Sacra to Velia. Okay. Levels of fluctuating asymmetry were very low for both of these populations. There was 182 distances calculated on the skull here, and out of the 182, only 32 of them were significant, so that's a pretty small percentage. Isola Sacra showed more asymmetry in about 2% of the distances, so they were more asymmetric 2% of the time. Well, Velia was more asymmetric than Isola Sacra about 16% of the time, so they did show more asymmetry, and that confirmed to our expectations. What this slide is showing you is the landmark distances that showed the highest and most significant asymmetric values for each group. What you notice is in red here, that's where we see more examples of fluctuating asymmetry in Isola Sacra. So the dotted lines, don't let those distress you. It's just that these are straight distances. So if you're going from the top of the head to the bottom, it's not going around the curve of the skull. It's taking it straight, right, in an XYZ dimension. So the dotted lines indicate it's kind of going through for that distance. But what we see is all three of these lines actually center on this top of the head in one straight point. But in Velia, we see some different endpoints. Most commonly, we see things more on the side of the skull, in the region of the cheekbone, around here. This is the region kind of underneath the cheekbone. You're looking at an inferior view or bottom view of the skull, and you'll see that it goes out to the palate, along again here on the cheekbone underneath, towards the back of the cranium. So something's happening in the side of the head, and it's changing the dimensions. Now, I've drawn these on the left, but it doesn't mean the left side was more asymmetric. It just means asymmetry occurred. Okay? And it could be the left or the right since it's random. So we were correct in our prediction that Velia would show more asymmetry than Isola Sacra. Then we compared Isola Sacra to Castel Malnome. What we see here again, levels of asymmetry were very low in both of these populations. Out of the 182 possible distances, only 27 were significant, so a very small percentage. What was interesting, and what really threw us, and we had a lot of talking about this, 18 of the distances, about 10% of them, showed more asymmetry in Isola Sacra compared to Castel Malnome, but only 9 distances were more asymmetric in Castel Malnome. That's not at all what we expected, right? We expected that Castel Malnome, because they're heavy laborers, they would be more asymmetric. And we saw the exact opposite thing. When we compared these two, it was Isola Sacra that was more asymmetric. So it's the same location in Isola Sacra. You can see the red. There's a couple extra lines here showing that there was more dimensions. But basically going to the bottom of the skull and the front of the face and the side of the head, all again centered to that point on the top of the head. And again, it's the temple region here, shown from underneath the cheekbone, going down to these points along the base of the skull. This large hole that you're seeing here, that's the foramen magnum, which is basically where your brain and spinal cord are connected and where that passes through. So overall, Velia displays greater amounts of fluctuating asymmetry than Isola Sacra, as we predicted. We were very happy about that. And this supports the idea that the individuals buried at Isola Sacra were healthier and suffered less developmental stress than those buried at that more southern port in Velia, down in southern Italy. These findings also support previous research showing a dietary difference between the two regions. The higher quality protein resources available at Isola Sacra may have made the difference. The better nutrition may have strengthened the individual's buffering systems to the stress that was happening. As I mentioned before, we know very little about the inhabitants of Castel Malnome, so we expected, due to the fact that they're heavy laborers, they're off the main trade routes to Rome, we thought that they would suffer a poorer diet and more disease than the coastal neighbors down there at Isola Sacra. These expectations were not borne out in this analysis. So overall, levels of fluctuating asymmetry were very low for all three populations. This suggests that severe or significant or severe environmental or nutritional stresses may not have been a significant factor in Imperial Roman times. Basically, life might not have been too rough for the people at this time. The middle-class lifestyles, the access to trade goods, the resources experienced along the two coastal towns may have allowed them to have a higher standard of health. And really, even with Castel Malnome, despite the fact that it's off the beaten track, it is still in very close proximity to Rome. So it's very well could have been trading with the large city. Also, we can't discount that these people were very likely farmers, which is what is the case for most rural populations at this time. They were raising their own crops and had what they needed to eat, and then could send the rest on for trade. Additionally, the second thing there is the strong canalization. Remember, this is your individual genetic component, your ability to withstand the environmental stress, your own body's ability to maintain that symmetric trajectory, and that tends to be genetic. So it's possible that strong canalization produced a consistent physical form or shape, despite all this noise that was coming at the people. It may have acted as a stabilizing force to maintain developmental integrity. An easy way to say that, maybe they just had good genetics. They just had good genes. There we go. One of the other advantages of this approach is the ability to identify regions of the cranium that are more or less subject to this developmental instability. We did find, if you remember from those pictures, that some regions were more likely to exhibit asymmetrical changes than others. We did see it around the front of the nose, the anterior part of the face there. Also along the palate, the roof of the mouth, those are regions more susceptible to fluctuating asymmetry. Other susceptible regions were along suture intersections, or where two or more cranial bones come together. We saw that especially at the top of the head in Isola Sacra, and a lot going on around here on the side, in the region, especially around the temple. So some regions of the skull, though, were very highly preserved. They showed little or no fluctuating asymmetry. And especially that was at the base of the skull where we have a lot of holes, what we call foramina. And in these holes, basically, you've got a lot of blood vessels and a lot of nerves passing through from the brain to the rest of the body. You're not seeing any asymmetry there. So we're thinking that this is a very genetically conserved region. If there's going to be asymmetry, it's not going to manifest where these critical vessels and nerves are passing through. It's going to manifest somewhere else. It's okay to have a little crooked, dimensions on the side or a cheekbone that's a little off. It's much more critical not to have those changes at the base of the skull. And indeed, the body is having very strong genetic control there. The ability to localize where these regions are, where they're being conserved and where instability is more likely to occur, is very relevant for a lot of research, human evolution, adaptation, along with growth and development. What I'd like to do in these last couple slides here is take a little twist and talk about something called consilience. And I think that this relates to the importance of the Center for STEM Excellence and the significance of the efforts on behalf of the natural sciences community here at the university and at universities across the country to reach out to broader communities and to reach out to social sciences and the humanities. I myself was very honored to be invited to speak here today because I'm a member of the Anthropology department and I'm not in natural sciences. And I was honored to know that they felt that I had something to offer you. This idea is beautifully detailed in this book. I've scanned the cover for you. Edward Wilson is a famous evolutionary biologist, and he wrote this book called Consilience. This is around the late 90s, I believe, and it's a book that the layperson could read. It's not a heavy scientific text. In it, he defines consilience as the linking of facts and fact-based theory across disciplines to create a common groundwork of explanation. And the origins for this kind of thinking were actually in the Enlightenment and the Renaissance. Wilson states in the book, and I quote here, "The greatest enterprise of the mind has been and always will be the attempted linkage of the sciences and the humanities. The biological sciences in particular have become consilient during the last 50 years. The basic divisions of biology are organized hierarchically. You've got evolutionary biology, ecology, organismic biology, cellular biology, molecular biology, and biochemistry. And the principles of each of these divisions can be telescoped into those of the others. This is a high degree of consilience or unity of knowledge. On the other hand, you've got other disciplines which are quite holistic, and that would include my own discipline of anthropology. And when you have a holistic approach, you tend to get a lot of people that specialize very narrowly and don't talk to each other very much. This increases complexity, and it's actually a great obstacle to consilience by synthesis. In the book, Wilson argues, and I think many of us would here agree, I certainly do, every college student, public intellectual, and political leader should be able to answer the question, okay, what is the relationship between science and the humanities, and how is it important for human welfare? A significant portion of the legislation coming before the United States Congress contains important scientific and technological components. Think of the own DNA that you may have to judge in court, or voting on stem cell research, and things like that. Most of the issues that challenge humanity daily, whether they be ethnic conflict, overpopulation, environmental degradation, or endemic poverty, these cannot be solved without integrating knowledge from the natural sciences with that of the social sciences and the humanities. Only integration across the boundaries of knowledge will provide solutions to these problems by allowing a clear view of the world as it really is, and not seen through the lens of these specialized ideologies or these narrow response to our immediate need. How many times have we seen environmental legislation go down the tubes right? Because, we want to get the taxes from the companies, okay? We don't want them to move overseas. So we can all breathe in toxins as long as they're employing people. And then you're going to have fluctuating asymmetry. So unfortunately, the vast majority of our political leaders are trained exclusively in the social sciences and humanities and have little or no knowledge of the natural sciences. Many consider human nature to be within their realm, and they have difficulty conceiving of the relevance of natural sciences to social behavior or to social policy. And on the other hand, to pick on the natural sciences, many of them have expertise that focuses very narrowly on narrow specialties, and they have very little connection to human affairs, and they're just as ill-prepared to engage any of these same subjects. Most American colleges and universities have diluted their curricula into a mess of minor disciplines and specialized courses. As we know, the number of undergraduates has risen exponentially. The percentage of mandatory courses in general education has dropped by more than half. We're losing the universe in the university. Only one-third of colleges and universities require students to take at least one course in the natural sciences. In education, the search for consilience may be the way to renew the crumbling structure of the liberal arts. That's the area that's usually on the chopping block when there comes to budget cuts. And thinking from my own perspective as a social scientist, rarely, if ever, do social scientists embed their narratives in the physical sciences, such as human biology or psychology, even though we know for sure that it's there, right, in the physical sciences and not some astral plane by which culture has arisen. We know that virtually all human behavior is transmitted by culture. We also know that biology has an important effect on the origin of culture and its transmission. The question remaining is how biology and culture interact, and in particular, how they interact across all societies to create the commonalities of human nature. The social sciences and the humanities, Wilson would argue, and I would agree, they can only benefit from an alliance with the natural sciences. Nothing fundamental separates the course of human history from the course of physical history. Researchers from both sides of the field need to come together and unite these great branches of learning. There has never been a better time for collaboration between biology, the social sciences, and the humanities, approaching basically a new age of synthesis. I feel that part of the STEM initiative allows us to move towards accomplishing this goal. We are reaching out to the local and broader community, bringing scientific advancement to the greater whole through things like this public lecture. While it may lure, as we hope, new practitioners to the natural sciences, we would like more students to study the natural sciences and major in these fields, it also brings an understanding of the natural sciences to the broader community, where it is very desperately needed. In ending, here we go, I'd like to thank the following people and organizations for their support and assistance. I am very honored to have been given the opportunity to come and talk to you this evening, and I hope it was enjoyable. And I especially would like to acknowledge you for coming and listening. So thank you very much. So they have told me that I have a few moments, I guess, for questions. Right up front, yes.
And do you think that your samples are typical of the population? What I'm getting at is, for instance, if your samples would be more likely to be preserved as. So do you think it's a representative?
Excellent question. I specifically didn't put my sample sizes up there. I will confess, this is a preliminary study. The work is preliminary. My samples are actually much larger. I had about 65 skulls in these samples here, so this is actually quite small. We are hopefully, this summer, we'll have more time to do more data analysis. Same thing, but on larger populations. And I do think that the samples are quite representative. The preservation is quite good. That being said, quite good, they're still 2,000 years old. So even though, I was told, oh, we have 500 skulls, when I got there was only 100 that I could actually measure because of the amount of fracturing and such. So the issue wasn't so much versus the rich or the poor, it was a lot of individuals from this time are cremated. And obviously, can't do anything there, and I actually showed the one slide where you saw the little cubby holes, I guess, in the wall where urns would be placed for cremated remains. That reduces sample size more than actual status differences.
Why were these ones saved when they were?
That's a good question. I've done a little bit of reading, but I have to confess I'm a little bit rusty. Maybe there's another person out there who would know a little bit about who gets cremated versus who does not get cremated. Yeah, I have come across that in my reading, but I confess it's been a couple years, and I don't remember the answer. Anything else? Oh, yeah, question way back there.
It's known that in Rome, you said you studied a middle class, middle class people, but a large percentage of the people populating Rome are slaves.
Yes.
And would you expect to see differences in the population?
Not necessarily. I mean, slaves were generally treated reasonably well. I mean, and often slaves could get their own liberty. And a lot of one site I didn't include here today actually had a lot of freed slaves and war veterans, and they were kind of doing a thing very similar to Castel Malnome, a rural site where you had a lot of farming and such. We don't believe that Castel Malnome was a large a property owned by a rich person who would have had a lot of slaves working the land. We actually believe it was a free settlement. And then Isola, Sacra, Velia, coastal ports, probably a lot of slaves coming in and out, but to the percentage that we see that might have actually been working those positions, it's hard to say. There's been some work done on something called genetic distance, and it does look like these populations were actually pretty homogeneous, which is actually very important for asymmetric studies. Because if you had a very heterogeneous, all people from different genetic backgrounds, it would be much harder to say what this asymmetry means, because you'd say, well, genetically, we don't know what their backgrounds were. But since the populations have been shown to be homogeneous from some other studies, it indicates people that had been there for a long time, and probably limited slaves, at least in these samples. It's a good question. I didn't expect that one. Anybody else? Yes, way over there.
How long before Castel data is going to include the environmental diet? And when you revisit our data at that time?
I would absolutely love. How long before Castel Malnome we'll have any information on diet? It could be an extremely long time. My experience with Italy has been there are so many skeletons coming out of the ground, they don't know what to do with all of them. Oftentimes they were cleaning these because I wanted to study them. They were along, in the caboose of the train here in the line of things to be cleaned. This was actually, that site was handled by what would be the equivalent of a CRM firm, a cultural resources management firm, so it wasn't at a university. That means it's much less likely to be studied. It's probably going to be cleaned, a basic inventory, some basic stuff, and I wouldn't be surprised if it's never published on. Once I publish the work and people see, they may try to access it a little bit more often, and so then more things may appear. I'd be fascinated. If they did go back and do the chemical isotope stuff, I would love to take another look at the data and see what the explanation may be, along with more reading in the humanities to see what might have been going on in the rural countrysides at this time, telling me more about the lifestyles of the people. So absolutely. But yeah, my hopes are not good for further publications, and there may be a couple that peek out. Oftentimes they're also published in Italian, of which I have beginner fluency, but I can do some reading, but it doesn't make most of the American audiences for that reason. So yes, up front. Right. Are there any plans to do facial reconstructions? None that I'm aware of. I don't actually do that work. Generally, it's people coming out of art or physical anthropologists that can do more than stick people, which is about the limit of my artistic ability. But it would be fascinating. I mean, we have the busts and the casts in the museums that were done of, the ruling families, at least at the time. So we have a general idea of what they looked like. But the common man can't afford those, to have those done and put on their gravesite. So none that I know of, but I would love to see it. If they ever had as the forensic movement becomes bigger across Europe and you see more facial reconstruction coming through forensic anthropology, you may get some forensic anthropologists that are interested in that with a little idle time who think, well, wow, I could, rather than only looking at modern cases, go back and actually reconstruct some of these ancients. Mm-hmm. Anybody else? Way in the back, yes.
Are facial fluctuating asymmetry being studied for different time periods and different groups?
Is fluctuating asymmetry being studied in different time periods and different groups? It's been studied a lot in the dentition of a lot of different people from modern day, I mean, I can think of one study that was modern day rural Mexican children compared to urban Texan children, and that was a big surprise. The rural Mexican children had less asymmetry than the urban Texan children. The authors had a lot of fun trying to explain that one. So you see it in modern populations and clinical studies, also throughout anthropology, but it's usually postcranial. Most, meaning the rest of the body, not the skull, and most of that tends to be directional asymmetry. The fluctuating asymmetry is almost always dental work. So almost 90% of what you'll read is looking at the teeth, looking at, the left molar versus the right molar. And I actually have a graduate student who, for her master's thesis, is looking at fluctuating asymmetry within Velia, just Velia itself in the dentition, because that hadn't been studied yet. So applying it to the skull, literally, there have been 2 papers besides mine, and one was by my colleague Valerie De Leon. She looked at ancient Nubians from the Sudan and southern Egypt from 3 different time periods, early Christian and post-Christian eras. And another person looked at them in Native American Indians, the Arikara from South Dakota, but that work was never published. And that girl is at the University of South Florida, and she's actually on to some other work now, so it doesn't look like it's ever going to see print. So looking at it in the skull is very new, but dentally there's quite a bit of literature out there. Anything else? Anybody? Yes, up front. A techie question. Uh-oh. Yep. Yep. Yes. Without moving the skull, I mount it. So the question, techie, a very good one actually. This is a big one for the methods. I would have given this to an anthropology audience. But how did I hold the skull still so that I could measure the top and the bottom? Because if you're manipulating this robotic arm, you've got to be able to reach everywhere. There's 2 ways you can do it. One way is you can take a series of landmarks, usually in the midline, digitize them, measure everything, and then flip the skull, and digitize those again, and then merge them. That's a nightmare to try to get it to happen, at least for anthropologists like myself. What I did was very particular in the way I mounted the skull. I put it on a clay base, a lot of clay, so that it was up about 2 or 3 inches, and I mounted it on the back of the head, and I would make sure that it wouldn't wobble, tapping, and as soon as it was mounted very well and completely still, I would take the measurements. And you barely have to touch the digitizer so you don't knock it. And then I could reach all of my landmarks without actually having to move it, and that made life much more simple for me. So, and every once in a while, there's 34 landmarks, but actually I measured 52, and this is on the left and the right side, so you're actually measuring over 100. You get to about 97, and oops, it jiggles, and just delete the whole thing and start over again with the whole skull. And they all were actually, for observer error, were measured twice. Yeah, and then merged. So anything else? Anybody, other brave souls, or inquisitive people? I'd be happy to stick around if you would like to talk to me more one-on-one or small group. I'm happy to answer more questions and be of assistance. So I hope you enjoyed it. I did.